This is Jae Cheol Kim, head of APAC Technical Solutions Center. The topic I would like to discuss today is NeoPro, which is a novel protein source for monogastric animals and released this year. So the content includes introduction, and we'll discuss about the base protein source, which is Bacillus fermented soybean meal, and then functional amino acid blend, which is we included in the product, and then nucleotides, and then we will introduce some of the animal trials that we conducted with this new product. As an introduction, the NeoPro contains the fermented soybean meal as a base protein source, which contains low molecular weight peptide, and then very low anti-nutritional factors and very highly digestible low molecular weight peptides. And then it also includes some probiotics, so very highly digestible protein source as a base protein. But what we know that when we actually try to replace animal protein with plant protein source, there are some discrepancies in animal response because animal protein contains some of the immune molecules that boosting immunity of animals. So we decided to include some of the functional amino acid blend, which is containing some of the functionality in improving mucosal immunity and enhancing antioxidant capacity, and also blocking some of the inflammatory cell signaling in the intestinal tract. So that will be effectively replacing immune boosting function of animal protein, even if it is a plant protein source. Then next one, the biggest difference between animal protein and plant protein is the concentration of nucleotide in animal protein, which is not in the plant protein source. So we included the nucleotide to accelerate epithelial cell regeneration, and then it also provides some prebiotic effect with increasing feed palatability. So including all this functionality, we believe that this new protein source, NeoPro, is a new economic functional protein source that safely replaces animal protein source in swine and poultry. So one of the benefits using this NeoPro in over the animal protein source is the safety as well. As you can see in the left-hand side graph, NeoPro contains none of the heavy metals in the product, while fish meals and other animal protein source often contains a large amount of the heavy metals and also a large amount of the ash. So NeoPro contains a lower ash content and none of the heavy metals, which is a very safe source, so that we can actually ensure the final pork product or chicken product without any contamination with heavy metals. So I'm starting with the base protein meal, which is the Bacillus fermented soybean meal. When we actually deciding selection of strains, fermentation of a soybean meal, we decided to go with the Bacillus, which is much more difficult technically to ferment the Bacillus compared to Lactobacillus and Aspergillus. But the decision to ferment the soybean meal with the Bacillus was based on the superiority in the growth rate and then production of carbohydrate and proteins by Bacillus strains during the fermentation compared to Lactobacillus and Aspergillus. Because with that functionality, more superior in production of carbohydrate and proteins during the fermentation, Bacillus fermentation more effectively removing most of the anti-nutritional factors in the soybean meal, and therefore make this soy protein product much more digestible without any concentration of anti-nutritional factors. One of the protein-based anti-nutritional factors in the soybean meal is trypsin inhibitor. Trypsin inhibitor is known to inactivate the trypsin and chymotrypsin produced from the pancreas. And when they actually reducing protein digestion, then gut can sense that protein digestion is not completely happening, and they actually sending negative signal by increasing production of a hormone called GCK from L cell in the small intestine. This hormone actually sending negative signal or sending signal to the brain, and then asks them to produce more pancreatic juice, which contains more trypsin and chymotrypsin. Therefore, if you feed a high amount of nutrition inhibitor from the soybean meal that you're feeding, from right-hand side graph, you can see that increasing amount of nutrition inhibitor intake in this case is broilers. And that there's a linear increase in pancreas weight because animals trying to produce more trypsin and chymotrypsin and often end up with pancreas hypertrophy. Even if it increases pancreatic juice production, and there's a still reduction in nitrogen digestion and increasing in endogenous nitrogen loss because they're producing more enzymes. And therefore, there was an increasing amount of non-digested nitrogen entering into the large intestine. That non-digested nitrogen now being fermented by microbiota in the large intestine and producing toxic gases by fermentation process of the nitrogen by microbiota, which is something not we want to happening in the gastrointestinal tract. Then next one is the graph that we can usually see the differences of the amino acid digestion between non-heat processed and then heat processed soybean, which mainly the difference is the concentration of anti-nutritional factors. For example, in here, feeding low flake to saccharomyces luster and measuring ileal digestibility, you can see that amino acid digestion is around 50 to 60%, which is very, very low. Because this low flake contains a trypsin inhibitor in there with some other anti-nutritional factors as well. But when you properly removing those anti-nutritional factors by appropriate cooking or heat processing, you can see that amino acid digestion is increasing up to 85 to 90% range. So you can see the differences of ileal digestibility of amino acid by presence of anti-nutritional factors in the soybean meal. And that's the importance of removing anti-nutritional factors in soybean meal to make them available for animals. Then we actually compare some of the competitor fermented soybean meal in here compared to our Bacillus fermented soybean meal. So competitor soybean meal in here mostly are lactobacillus fermented or enzyme treated product. While they only contains about 30%, up to 30 to 38% of the low molecular weight peptide concentration, Bacillus fermented soybean meal contains much higher low molecular weight peptide concentration, which indicates that how effectively that Bacillus producing proteins during the fermentation and therefore reducing the chain length of the peptide in the soybean meal. Because of that, Bacillus fermentation is known to break down more trypsin inhibitor compared to other fermentation methods. For example, in here, you can see that the concentration of allergens and then trypsin inhibitor in the soybean meal product or lactobacillus fermented soybean meal, LFSBM is lactobacillus fermented soybean meal, or ET soybean meal, which is enzyme treated soybean meal, compared to those products, Bacillus fermented soybean meal, more effectively removing most of the protein-based anti-nutritional factors such as allergens and trypsin inhibitor. Then there's the carbohydrate-based anti-nutritional factors in the soybean meal. And most important carbohydrate-based anti-nutritional factors are galacto-oligosaccharides and also major galacto-oligosaccharides in soybean meal stachyose and lactose. And if you see in here, left-hand side, the broiler studies, increasing stachyose content from 1 to 3%, you can see that there's a linear reduction in total ME content in the diet, so that energy extraction from the feed are significantly reduced by inclusion of 2 to 3% of stachyose in diet. On the right-hand side, when it was tested with the 12 kilogram pigs, inclusion about 2% of the stachyose significantly reduced every daily gain in nitrogen retention. So galacto-oligosaccharides in monogastrics are significant negative nutritional factors for swine and poultry production. If you look at this HPLC analysis on the left-hand side, soybean meal, you can see that there's a large peak of lactose and stachyose content. But you can see that lactobacillus fermented soybean meal can reduce some of the lactose and stachyose, but not completely. But Bacillus fermentation almost completely removing lactose and stachyose content. So right-hand side, if you see that in the percentage reduction from soybean meal, you can see that stachyose and lactose content is quite large, like a 4% and 1.8%. And that was almost completely removed by fermentation of soybean meal with Bacillus subtilis. Then Bacillus fermented soybean meal actually contains Bacillus itself as a probiotics. For example, in the left-hand side, you can see that Bacillus is highly heat stable. So at 100 degrees, there's no reduction up to 30-minute cooking. At 120 degrees, there's a slight reduction in its concentration when it was cooked up to 30 minutes. Compared to the diet, lactobacillus is not heat stable at all. So at 45 degrees, most of the lactobacillus is disappeared. So what it means is that Bacillus fermented soybean meal contains its Bacillus as a probiotics, and it will be stable in the pelleting and extrusion process. So we did one study that with the extruding test of the shrimp feed, which include about 10% of our base proteins, Bacillus fermented soybean meal. And then you can see that up to 110 degrees, there was no reduction in Bacillus number. At 120 degrees, there was a one log reduction of the Bacillus. So most of the pelleting process, this Bacillus will survive and then will provide action as a probiotics in the feed. So we did some testing again in vitro. In the left-hand side, looking at the antibacterial activity with the E. coli and the Salmonella for the livestock strains, and then for aquaculture strains like Vibrio and Aeonomas. We actually added about 10 to the 5 to 6 Bacillus culture medium on the flask that growing these pathogens. And you can see that with the Bacillus culture medium, these pathogenic bacterial growth is diminished. So it has a highly antibacterial activity. On the right-hand side, we also tested the quorum quenching effect. So if you look at the negative control, the blue mark is the quorum chemical production by pathogenic bacteria. And as you can see with this 1, 2, 3, 4 Bacillus medium dropped in there, you can see that quorum chemical production was significantly reduced. So this Bacillus that is included in the base protein will reduce the chemical communication between pathogenic bacteria. Therefore, it will prevent the increasing virulence factor of these pathogenic bacteria in the gastrointestinal tract. Another benefit of using Bacillus fermented soybean meal is that Bacillus is actually surviving in the fishes as well. So when they actually testing these fecal material for 7-day fermentation, you can see that ammonia production in the fishes are also significantly reduced. So about 30% reduction in ammonia production in the waste product, which means that if you use this product, Bacillus will reduce ammonia production in the farm levels. So that will be contributing to reduction of the more problem with ammonia in the farm for the human and also for the animal as well. Now, on top of that, we decided to add some functional amino acid blend because we know that plant protein source usually doesn't have any functionality in improving immunity, stimulating immune function of the animal compared to animal protein, which contains mostly immunoglobulins and other immune stimulating molecules. So if you look at this research data, that many individual amino acids actually act as a signaling molecule inhibitor. For example, methionine blocking production of nucleotide kappa B, which is increasing intestinal inflammation. And then tryptophan is blocking MAPK production, which is also increasing intestinal inflammation, or other amino acid also involved in many signaling molecules that associated with intestinal inflammation. So proper blending of this amino acid and supplementation in the feed will improve immunity of the animal, and therefore mimicking the similar effect of immune boosting effect of the animal protein, even if we are using soybean plant protein source. So we did in vitro test with the pig's primary macrophage cells, and then we stressed these macrophage cells with LPS, which is endotoxin from E. coli, and then induced inflammation response. And then we analyzed these samples with RT-PCR for inflammatory cytokines. And you can see that some amino acids like methionine and threonine increased anti-inflammatory cytokine production, while tryptophan and arginine decreased pro-inflammatory cytokine production. So, proper blend of these amino acids together and supplementation will definitely increase immune defense system in the gastrointestinal tract or monogastric animals. So if you look at this concentration of functional amino acid content between our new protein source NeoPro and compared to fish meal or DPS, which is the hydrolyzed intestinal cell protein, you can see that there's about double the amount or 2.5-fold higher functional amino acid content in NeoPro compared to other animal protein source. These functional amino acid is not going to be included as a matrix in the formulation because we're not giving out this additional functional amino acid blend concentration as a matrix. So these functional amino acids will be going on top of your amino acid formulation. Therefore, they will provide extra immunity to animals. The decision based on that inclusion of functional amino acid blend was based on the very recent studies that actually showing that this addition of a functional amino acid blend, in this case methionine, tryptophan, and threonine, significantly improving performance of the pigs after infection with the salmonella. For example, in here, whether it's low protein and high protein, it doesn't matter. If you supplement this functional amino acid blend, there was a significant improvement in everyday gain and feed conversion efficiency 7-day post-inoculation with salmonella, which indicates that how functional these amino acid blends are for the bacterial infection. And then secondly, from the same group from the Canada, they actually found that including functional amino acid blend for the 2 weeks before the actual event of the salmonella inflammation, there was a significant reduction in colonic salmonella proliferation. For example, in left-hand side, FAA minus means not including functional amino acid blend. And FAA plus 0 is including functional amino acid blend on the day of infection. FAA plus 1 and 2 is including functional amino acid blend 1 to 2 weeks before actual inflammation with salmonella. You can see that by supplementing functional amino acid blend 2 weeks before actual event of the inflammation with salmonella, there was about 50% reduction in salmonella content in the colonic content, which indicates that somehow this functional amino acid blend decrease the proliferation of salmonella. And the right-hand side, you can see that performance response of the pigs. And then again, if you feed these functional amino acids 2 weeks before actual inflammation, there was a significant improvement in everyday gain. So functional amino acid blend, which included in this NeoPro product, if you feed them before 2 weeks, 2 weeks before the inflammation, then there was a significant protection effect in the gastrointestinal infection from pathogenic bacteria. And there was another study from South Dakota University in 2022. They actually used the functional amino acid blend, including tryptophan, methionine, threonine, arginine, histidine. And then they found that during the study, unfortunately, they had a natural infection with a PRRS. during the 8 weeks period, but by supplementing functional amino acid blend, there was a significant improvement in FGR, which indicates that functional amino acid blend somehow also works for the viral infection as well. And then in this study, there was a significant improvement in livability of the pigs. We need to be careful about this interpretation of this result because this is very small scale study. So the actual scale of the livability will in the commercial farm will maybe very different. But the major message of this study is that the functional amino acid blend actually have some effect in viral disease as well. The next shortcomings of the plant protein source compared to animal protein source is that the animal protein source contains large amount of nucleotides while plant protein is not. Nucleotide is essential for rapid regeneration of the gastrointestinal tract. For example, in the right-hand side, you can see that the crypt contains the stem cells, and these stem cells are produced by mitosis and then differentiated and migrate and maturated up to the fetus tip. But what happens when animals are infected with pathogenic bacteria or there's other viral infection? There will be intestinal damage. Therefore, villus will be shortened, and then crypts will be getting deeper because animals start to producing more cells and increasing mitosis, and they need rapid regeneration of enterocyte, and they need nucleotides. So if you just feed them plant protein source without including any nucleotide, and then removing animal protein, there will be certainly a slowdown of this mitosis, and therefore regeneration of epithelial layer will be compromised. If you look at these differences between fish meal and soybean meal nucleotide concentration, the major nucleotide that is deficient in plant protein source compared to animal protein source is IMP. So actually included the IMP in the nucleotide to compensate those reduction in nucleotide in plant protein source. So NeoPro is a complete protein source that designed to replace animal protein, and it also will be a very safer source and also more economical source to replace animal protein. So we have some animal trial result in here. So we're gonna share only a few of them. We have much more animal trial data. So if you need more details, then you can contact us. But we'll show you some of the animal trial results in here. The first trial we conducted was the university trial to measure the energy content and amino acid digestibility in vivo. So in here, you can see that we use the soybean meal 48% and soy tart 53%, which is our Bacillus fermented soybean meal, and NeoPro with the functional amino acid blend plus nucleotide inclusion in the product. And then we also use the one competitor product, which with the fermented soybean meal product. Then we measured the metabolizable energy content compared to soybean meal or competitor product. You can see that soy tart, our NeoPro product, contains about 10% more metabolizable energy, which is another source of economic benefit that you can actually implement in your formulation. You know that energy is the most important nutrient and most expensive nutrient in the formulation. By using this NeoPro, you can actually save some energy as well. And then secondly, we measured standardized ileal digestible amino acid content. If you look at in here, left-hand side, indispensable amino acid and dispensable amino acid are very similar response. And then right-hand side, the total amino acid, standardized ileal digestible amino acid, digestibility of the amino acid are very different from soybean meal and competitor product with the NeoPro, which is about 5% higher compared to soybean meal product. So you can see how digestible these amino acids are in NeoPro product compared to soybean meal or other fermented soybean meal product. We did one study in Vietnam University, Nong Nam University. We used the 72 willing pigs. We had three different treatment. The control, which is a standard diet with a 4% of the fish meal, 1% of the plasma protein, and 2% of the hydrolyzed intestinal protein source. The NeoPro only contained 1% fish meal and then no plasma, no hydrolyzed intestinal protein, and then 5% inclusion of NeoPro. And the competitor product, we added 2.5% fish meal and 0.5% of the plasma and 1% of the hydrolyzed intestinal protein source, and then 5% of the competitor fermented soybean meal product. And you can see that every daily gain, while competitor product is not improving, NeoPro improved every daily gain by 35%, and then reduced FCR by 9%. Significant difference between other fermented soy products in the market. From the same trial, we actually looked at the concentration of the E. coli and Bacillus, and then lactobacillus content in the gastrointestinal tract content. You can see that NeoPro increasing Bacillus and lactobacillus while decreasing E. coli concentration, while competitor product is not changing much of the E. coli, Bacillus, and lactobacillus concentration. Because of that, NeoPro reduced diarrhea compared to control diet by around half, 50%. And then also, if you look at this intestinal structure, jejunum was much longer, crypt was much deeper, and then jejunal villus height was much higher. So villus height to crypt depth ratio was much better than control and competitor product, which indicates that it actually improving gut regeneration much better than other competitor product. And then from this trial, we actually did some economic analysis. And then you can see that by using NeoPro, there was a reduction in feed price. By multiplying feed intake, there was about $2.3 less per pig of feed cost. And then there was about $1 more benefit by increasing weight with the pigs. So in total, from this trial, there was about $3.3 benefit per pig by using NeoPro compared to animal protein in the control, while competitor product is not improving much of the economic values. And then there was another trial that was more like a farm field trial. And this farm actually had a commercial diet, including 5% of the fermented soybean meal. And then we replaced this 5% of the fermented soybean meal with a NeoPro, and then measured or compared the performance in here. What we found was that by replacing 5% of the fermented soybean meal with a NeoPro, there was about 14% improvement in final body weight. And also there was increasing everyday feeding, daily feed intake, and about 5% improvement in FCR in this trial. Looking at the economic benefit in this trial, there was an increase in feed intake by replacing conventional fermented soybean meal with a NeoPro, but because of the huge differences in live weight at the end of the trial, like there was about 2.5 kilo differences at the end of the trial. Therefore, about $6.5 differences in live hog price. And in the end, considering that increase in feed cost, there was still about $4 benefit per pig by using NeoPro in this conventional feed. So that was all. So if I conclude this presentation, comparative analysis showed that NeoPro contains much less anti-nutritional factors and heavy metals compared with animal protein source, which indicate that which is much safer and then more digestible protein source. And NeoPro shows better digestibility than soybean meal and fish meal, confirmed by swine in vivo and in vitro digestion trial. And then thirdly, NeoPro provides various functions, reducing inflammation, oxidation, and heat stress due to optimal amino acid and IMP inclusion. And therefore, this novel functional protein source called NeoPro can safely replace animal protein source. We are improving feed safety, nutrient digestibility, and immune function of young and breeding animals. And it will be a very effective and safe protein source for the future animal production. So with that, thank you very much for your attention. And then if you have any questions about this NeoPro, please don't hesitate to contact one of our representative in your region. Thank you very much.